Hello and welcome to section 2 on our chapter 21 video lecture series. In this section, we'll talk about barrier defenses and innate immune responses. First, let's do a quick little background. Our immune system, we know that we've got two immune systems, our innate immune response, which is going to be non-specific, so we'll have some physical barriers in here. It's going to be rapid and not always as effective as our adaptive immune response. So it'll take more time because we are going to tailor make a defense. It's highly specific and effective. And here's a little schematic to talk about those two different systems. You could see within innate, we have our physical barriers and our blood-borne pathways, such as our complement system and phagocytosis, and we'll also have inflammation within this route. Our acquired or adaptive immune system will consist of our B-cell immunity, also known as humoral immunity, and T-cell immunity, known as cell-mediated immunity. And here's a little table that will walk you through the barrier defenses. This would be within our first line of defense. So our skin is, of course, being a physical barrier. We've got our keratinized cells that create a surface, and also our Langerhans cells that are going to work within our immune system. Our skin also consists of sweat and secretions such as oil secretions. So that we would use our sweat glands and sebaceous glands to produce, and that then is going to give us the result of a low pH acidic mantle on our skin, as well as a washing action. Our oral cavity has the salivary glands within it, which contain a chemical known as lysozyme that is going to help inhibit bacteria. Our stomach within our GI tract is very acidic, so we've got low pH there to combat pathogens that cannot withstand that pH of 2. Within our mucosal surfaces, we are going to have that lining our GI tract and respiratory tract, and there we have non-keratinized epithelial cells to protect us. And lastly, we have our normal flora, or good bacteria, as most people know it, and that can be found through our mucosal tissues and can basically take up real estate, so it can prevent the pathogens from growing on those mucosal surfaces. Next, let's talk about some of the cellular responses in more detail within our innate immune system. So we've talked about phagocytosis a bit, and this is when we go through that process of endocytosing the pathogen. In this case, it looks like it's a bacterial cell, and this is done by our neutrophils and macrophages. So here we can see this is a macrophage that's taking up the pathogens, but of course we'll look at this cartoon version here first. So as we endocytose that bacterium, we are then going to place it into a phagosome. That phagosome is then going to fuse with a lysosome that has the digestive enzymes within it that kills the bacteria cell, and now we've got all these little cellular components that we need to get rid of, so it exocytoses it from the cell. Now, sometimes it will take antigens and place them on MHC molecules so it could present it to other immune cells. Note that our monocytes, which in the blood we really call them monocytes, but as they move into the tissues we call them macrophages, but these monocytes can differentiate into dendritic cells, and that serves as an antigen presenting cell too. So let me give you a quick little list here. Our antigen presenting cells would consist of dendritic cells, macrophages, as well as our B cells. Now let's move into our neutrophils. Our neutrophils are our white blood cells. They classify as granulocytes because they've got those large granules, and they are phagocytic cells that are typically the first to arrive when we've got some tissue infected. They only last for a few hours, and then the macrophages are going to come in. So they'll do things like cross the wall of our GI tract to help provide protection. But like we said, it just lasts a few hours. Then our macrophages come in second. These are very large phagocytic cells. And as I said before, our monocytes are traveling through the blood. And once they enter the tissues, we now call them macrophages. They are going to be longer lived and can ingest larger particles. Sometimes they take up residence in specific organs. So for instance, in the liver, we call them Kupffer cells. And we also have specific macrophages called histiocytes, which are found in connective tissue, and alveolar macrophages that we find within the lungs, or these cells have also been nicknamed, one of my favorites, called dust cells, where they go around and they eat up debris in the lungs. Now these phagocytes and our cytotoxic NK cells that we talked about in our previous section are going to recognize patterns of pathogen-specific molecules, and that consists of PPR, pattern recognition receptors. These are membrane-bound receptors, which you could see illustrated here, that are going to recognize characteristic features of our pathogens that are released by stressed or damaged cells. So this is gram-negative bacteria, and one of their specific characteristics is within their cell wall, they have something called LPS, lipopolysaccharides, which our cell here is recognizing and binding to and can activate a signaling pathway that will then cause our cell to go through transcription and release NF-kappa B to enter the nucleus. And by entering that nucleus, we are now able to secrete cytokines such as IL-8, IL-1, and TNF-alpha, tissue necrosis factor alpha, which are then going to go and continue the pathway for our immune response. So you may have heard me use the terms cytokine or chemokine already. Let's differentiate what those terms mean. A cytokine is going to be a signaling molecule that's allowing the cells to communicate with each other over short distances. So the cells are not far from one another. Our chemokines are going to be soluble chemical mediators that are similar to our cytokines, except for its function to attract cells. And that function to attract cells is called chemotaxis. So I just think chemo for chemical, taxis for taxis, like we're moving in a certain direction. So this takes place over long distances. So we are able to release the chemicals, and then our cell is attracted to that and will move closer to it. Next, let's talk about interferons. These really play a role in preventing viral replication. So what happens is we have our cell here that unfortunately gets infected with a virus. Now, as that virus is hijacking our machinery of our cell to create viral parts and assemble them, and those viruses then leave the cell to try and attack another cell. Meanwhile, what happens is inside our nucleus, we are going to have our interferon gene activated. And when we activate it, our interferons are going to leave that infected cell and go to our neighboring cells and gives it a warning. It signals the nucleus to turn on its antiviral proteins so that we can combat that virus from being able to replicate inside the cell. So we're destroying those particles, and it puts a stop to the virus. Next, we have opsonization, and this is a function that could be done through our complement proteins or our antibodies. And essentially what we're doing here, you could see our antibodies illustrated. They help to tag the pathogen so that it marks it for destruction through things like phagocytosis. So our phagocyte cell recognizes this complex with our antibodies and then will grab it in order to eat it up. Here's our complement system that I've been mentioning here and there. So our complement proteins are made up of 20 different proteins, and these proteins are named C1, C2, C3, I believe based on how they were discovered, and they tend to circulate within our blood in an inactive form. Once they become activated, they can go through different pathways. So we've got our alternative pathway, part of our innate immune system, where one of our complement proteins, C3, is going to bind with the foreign substance and attract macrophages. So essentially doing opsonization, where we're marking it for destruction. Our classical pathway is a part of our adaptive immunity, and this requires antibodies bound to the antigens. So essentially what's going to happen is we have our complement proteins work together to eventually form what is known as a MAC complex. MAC stands for membrane attack complex, which creates these pores or channels throughout the plasma membrane of the cell, which will allow all the fluid to move into the cell, causing it to lyse and killing the cell. We also talked about attaching to surfaces of the cell and marking it for destruction through phagocytosis. And we can attract our immune system cells to the site of infection to promote inflammation. And that's what's taking place in this illustration here. Please note that for this next slide, you do not have to memorize the pathway of these complement proteins, but I just wanted to give you a visual of how complex one of these pathways are. So here we have complement protein 1, so C1, which forms a complex with antibodies. And when it does that, we create an enzyme, and then it can travel over to C2 and C4 to create another complex that is then going to cleave C2 and C4 into C2A, C2B, and C4A and C4B. The C2A and C4B come together then to form an enzyme. That enzyme is then going to travel to bind with C3 and will cause C3 to cleave into C3A and C3B, which C3B then comes together with C2B and C4A and forms another enzyme. These will then, as an enzyme, travel and bind to C5, and C5 will then cleave into C5A and C5B. C5B then travels and binds to C8, C7, C6, and a lot of C9s, which then forms the MAC complex, that pore that is on the plasma membrane of the pathogen to allow fluid to move in and lyses the cell. Now let's talk about our innate immune response for inflammation. At this point, tissue injury, regardless of the type, can cause inflammation. Our response is initiated through chemical mediators that are going to cause things like vasodilation, chemotactic attraction, and vascular permeability. So let's take a look at that real quick in this image. We see that we have a wound, and unfortunately it's allowed this bacteria to move in. So at that point, we are going to have some mast cells release histamine and other mediators to attract more of our immune cells to come to the site, mainly our neutrophils first, right? They will also cause our vessel here to increase its permeability. So by increasing its permeability, we're able to allow the blood contents, such as our neutrophils, to be able to move on out. So this process is called leukocytosis, where we have the release of our neutrophils from the bone marrow in response to our leukocytosis inducing factors from the injured cells. Then what starts to happen is these neutrophils will start to migrate along the walls of the capillaries here. And there's certain receptors that kind of slow them down. Think of them as like speed bumps. And as it slows down, it finds a nice little space between the endothelial cells and goes through a process called diapedesis, where it's going to flatten and squeeze through the capillary wall. Then we have chemotaxis taking place, where the inflammatory chemicals that are being released are helping to attract it to this injured site. So our neutrophil here, you can see, is going through the process of phagocytosis, and then our macrophages are going to also arrive, gobbling up the pathogens or any larger particles, and can further release cytokines to continue to allow more immune cells to come to the site and initiate tissue repair. We can also call upon fibrinogen, and we, through our pathway, can convert fibrinogen over to fibrin, and this helps to create a wall. You could see that wall forming here with our blood platelets to separate out this injured site from the healthy tissue. Now, when we have inflammation, this could be local, like we saw in the previous slide, where we're going to have some cardinal signs, such as redness, and that would be from the vasodilation causing more blood flow to the area, which also will result in heat. Since we have more fluid in the area, we will also get swelling, and with that increased blood flow and swelling, that pinches our free nerve endings, which causes pain, and that tissue is no longer functional. So we have our last, fifth cardinal sign that was most recently added, known as loss of function. Now, when we have systemic infection taking place, it's in lots of parts of the body. So we have same symptoms as local inflammation, but we also have the addition of increased neutrophil numbers that are being released through our red bone marrow, a fever due to the production of pyrogens, and this fever helps to improve performance of our immune system. We'll also have widespread increased vascular permeability because histamines are being released throughout our body, and a large volume of plasma is going to enter our interstitial spaces, which can lead to shock.